be here in the ICMS 2026, and it's a great privilege to introduce Professor Matt Vijayan. I know him from quite long, from my PhD days. He is a professor in University of Calgary, and he is a Canada Research Tier Professor. And also he received several awards. Some of them are mentioned here. Premier Research Excellence Award from Province of Ontario, Bob Butler's Award for Canadian Society of Zoologists, University Research Chair of University of Waterloo, and he is a well-known environmental physiologist, and he is known for his work on corticosteroid signaling under stress responses and signaling in using fish model. And he has done some of remarkable publications, and also he has got several citations as well as H-Index 73. And he is also editor of several journals and also reviewer. And it's quite happy to have Professor Matt Vijayan here. And then, in fact, I took the liberty to invite him when I met him in the meeting in Vadora, and he had kindly agreed, and I now request Professor Matt Vijayan to be here on the stage. Thank you so much, sir.
Awesome. Thank you. Thank you so much. It's kind of surprising how I ended up here. I met Professor Senthil Kumaran in Vadodara, and he was kind enough to invite me to come and give a talk. And I should also thank Dr. Indrashis Bhattacharya for accommodating me at such a short notice. I'm just here for a day, unfortunately, so I'll be missing a lot of talks for tomorrow and the day after. My flight is on 2 days from now, so I have to leave tomorrow. Anyway, so Professor Balram gave a really good introduction about chemical biology and sensing, but he spoke mostly about peptide molecules, and this will be a slight deviation from that lecture, and I'm going to talk about corticosteroids, which are lipophilic steroid molecules, but the signaling component is exactly the same as any other molecule we can think of. So what I'm going to talk about is some of the work we did in my lab, and I'm going to show you some results, not very much. Mostly I'll try to talk about the concepts, but I do bring some of the results just to show how we got around doing some of the work. And the laser pointer is not working, so I think I'll just point it. I'm from University of Calgary, and if you have any questions, if you want to chat with me, especially, my talk is geared towards students, so people, if you know whatever I'm talking about, I'm sorry, but I'm getting more towards students so they can understand what I'm talking about. Yeah, so what I'm doing is I provide an outline. So essentially I'm going to go through the different ones. I'll first talk about stress response, and that's a very fundamental process that happens in every organism. So no matter what model I talk about, it's the same conserved phenomenon. And one of the key player here is the steroids. And so I'm going to talk about the steroid. And then the second aspect would be I'll talk about the receptor, like you heard earlier in the lecture, how important receptors are in communicating the signal and all the downstream effects that can happen after that. So I'll talk about the corticosteroid receptors. And the focus of my talk is going to be on a receptor called mineralocorticoid receptor, or MR, which is very common. It's got a lot of biomedical applications in humans. For instance, anything related to blood pressure or cardiac problem, you end up taking a medication that's related to MR. Either it will be antagonist or agonist, depending upon your condition. So I'll talk about the uniqueness of MR in the model that I'm using, and it's very different. So from an evolutionary standpoint, it provides some fundamental insights into what may have happened to this receptor and how some of these ligands evolved to communicate the signal. And then I'll talk about some, so I'll show you some results. I have to show some results. So I'll show you some effects we see in behavior associated with corticosteroid receptors. Then I'll talk about its role in metabolism and growth, and finally I'll conclude. So I'll try to limit my talk to sort of 40 minutes, and hopefully, I'll be able to do that. Maybe you can control it. No, use the first one. The first, no, that's fine, don't touch that. The next, yeah, that one, please. No? Oh, it's all okay. Is it working now? Okay, perfect. Awesome. Yeah, so I'll just set the stage for everybody. And like I said, the stress response is highly, highly conserved. And you probably know about the sympathetic nervous system where, again, peptides come into play. And that's a very rapid response, what you see. And normally you're associated with the fight or flight response. And I'm going to focus more on the hypothalamus, pituitary, adrenal axis. That's what we have. And regardless of whether it's a fish or a bird or an amphibian or you're all going to respond to a stress by eliciting a response which releases corticosteroids into the circulation, which is slower acting. Unlike the peptide hormones, these are more, they're lipophilic, they go into the cell and they activate a receptor, which is actually a transcription factor. Okay, so that's where it differs from the receptor that Professor Balram was talking about, which is more membrane-oriented. The peptides bind to the membrane receptor, elicits rapid signaling, mostly phosphorylation, dephosphorylation. So this is just to, again, put things in perspective. So normally what happens is you have a very rapid response in any type of stressor, and catecholamines go up within seconds to minutes, and it's also quickly turned over. So the half-life is very, very rapid. Whereas the corticosteroid response is slower acting and it lasts longer, okay? And that's what I'm going to focus on, is this longer acting effects and what is the significance of that. So this is essentially the hypothalamus, pituitary, adrenal axis that I'm going to talk about. And so as you all know, again, whether it's humans or animals, it's the same sort of pathway. The hypothalamus produces a hormone called corticotropin-releasing hormone, which is the primary stimulus. So any type of stressor stimulates neuronal cells in the hypothalamus to release corticotropin-releasing hormone, which acts on the pituitary to release another hormone called adrenocorticotropic hormone. That's a post-translational product, by the way. The gene that encodes it's a pro-opium melanocortin, or POMC. And the ACTH then binds to receptors, which are membrane receptors called MC2R, or melanocortin-2 receptor, on the adrenal cortex. And that's what's stimulating the whole steroid biosynthetic cascade, leading to the release of corticosteroids into the circulation. So that sort of pathway is highly, highly conserved, okay? And in animals, like in fish, I work with fish. I use fish as a model. They don't have a discrete adrenal gland, but they do have the same cell types. They have the same steroidogenic cells, which are distributed all along the kidney of a fish. And then the main players of my talk are these two receptors, which are the glucocorticoid receptor and the mineralocorticoid receptor. And they both are very, very important in our body as well as in most animals. And it's ubiquitously distributed. So every cell in the body have those two receptors, okay? And then once corticosteroid gets released, there is a negative feedback regulation that regulates the level of corticosteroids in the circulation. It can happen at the level of the hypothalamus, or it can act locally at the level of the adrenal cortex. So now I'll go to the corticosteroid receptors. So like I said, the glucocorticoids are lipophilic molecules. They go through the lipid bilayer membrane, and then they bind to a glucocorticoid receptor, which essentially becomes a transcription factor upon ligand binding. And there's a number of protein complexes that keeps the steroid receptor in a ligand binding conformation. I won't go into the details. I don't have the time for that. But there are a number of molecules involved. But once the ligand binds to the receptor, they form a homodimer. Okay, so two GR with the ligand binds together. They go into the nucleus and activate targeted genes. So those are the glucocorticoid responsive genes, and they play a whole slew of responses to the body, including iron, growth. I think I have it here. Development, stress adaptation, metabolism, immune regulation. when we think about glucocorticoid or corticosteroids in general, the immune regulation becomes the most important component. If one of the most widely dispensed pharmacological compound is prednisolone and corticosteroids. So anything related to asthma, inflammation, you use corticosteroids. And even during COVID, corticosteroid was one of the highly prescribed drug to prevent to cause immune suppression. Anyway, so it's a very important receptor, and it conveys a lot of messages to the body. And one such thing is how you cope with stress. And one of the things that happens with stress is there is an increased energy demand, and so you need resources to cope with this increased demand. And that's one of the jobs that this particular receptor does, is mobilizing energy resources. But then there is also another receptor, which is the mineralocorticoid receptor. And it's what this one does, at least in humans, is it's very important in iron regulation. It functions exactly the same way. The big difference is the mineralocorticoid receptor has a higher affinity for cortisol than for its endogenous ligand, which is aldosterone. So there is an enzyme called 11-beta hydroxysteroid dehydrogenase that breaks down cortisol, makes it inactive, so your aldosterone can actually bind to the receptor and target MR responsive genes. Okay, and those genes are very important, again, in development, metabolism, osmoregulation, and particularly iron regulation. So this is where I'm going to diverge a little bit from the model I'm going to be using and the significance of that. And in humans at least, the main role of MR, from a biomedical standpoint, one that's got a lot of attention, is its role in iron regulation, water control, because one of the biggest challenges we face is dehydration and water regulation. So you have this particular receptor playing a very important role. But what's really cool is the model I use, which is zebrafish, doesn't have aldosterone. We heard about the evolution of how new molecules evolve because of the demand or the need for it. And that's essentially what happened here. In fish, they don't have aldosterone. They lack the enzyme called aldosterone synthase. So they cannot produce aldosterone. So cortisol does both the job of your glucocorticoid and the mineralocorticoid, which is really interesting. That's why I use that as a model. So now from an evolutionary standpoint, we can ask the question, why did they diverge? Why did you have this aldosterone being synthesized in humans and in tetrapods, but not in other animal groups? So essentially, when we think about cortisol signaling, most of the studies you look at stress adaptation, they all focus on glucocorticoid receptor because we know a lot about what that receptor does. It's important for stress coping, development, growth, metabolism, immune response. But we know very little about what mineralocorticoid receptor does other than its role in iron regulation. So from a stress standpoint, the question in my lab was, what is the purpose of having these two receptors with one common ligand in a teleos model? And what was the reason for this evolution of aldosterone in tetrapods? So if you look at the phylogeny, you have a common ancestral receptor, which is the corticosteroid receptor. You see that in agnathans, in lampreys and hagfishes, etc. But then as you move up the scale, when you go to the elasmobranchs, it starts diverging. You have a glucocorticoid receptor and a mineralocorticoid receptor, and that continued all through to the tetrapod lineage. So the only difference is all these had corticosteroids like cortisol or 11-deoxycortisol, and that would bind to mineralocorticoid receptor. But once you come to the tetrapods, you see aldosterone being synthesized. So that's why the fish forms a really interesting model where we could ask this question, why did this happen? And what is the role of cortisol? And how do they regulate these two receptors distinctly or independently? So the mineralocorticoid receptor was discovered in 1987 by Ron Evans' group at Salk Institute. And in 2017, they had a symposium on the discovery of mineralocorticoid receptor. And in fish, MR was discovered in 2000. But what was really interesting was, in 2017 at this meeting, we still didn't know what MR was doing in fish. We had no idea. And the primary reason for that was MR did not have a really good antagonist. We used spironolactone or eplerenone, but they're not highly specific. So people were unable to study exactly what MR was doing in fish. So we decided, at that time I had a PhD student, and she was really interested in understanding what MR was doing in fish. So we decided to characterize the role of MR in fish. And so what we ended up doing was, the best way, if you don't have an antagonist, the best way to go about doing it is by knocking out the gene. So, and that was the time when CRISPR-Cas was out there, and it was very easy in zebrafish to knock out a gene. I say that because the zebrafish is a phenomenal model for biomedical research, because nearly 70 to 80% of the coding genes are similar to what we have in humans. And on top of that, all the disease-producing genes are all there in zebrafish, including mental disorders, you name it. So we can mimic Parkinson's disease, Alzheimer's disease sort of phenotype in zebrafish. So we decided to knock out both GR and MR, and then try to figure out exactly what this receptor is doing in fish. So that's essentially what we ended up doing. But what's really neat is, if you take a rodent model and you try to knock out GR or MR, they don't survive, right? Because the glucocorticoid receptor is very important for lung development. And mineralocorticoid receptor is very important for, like I mentioned before, for water conservation. So when you knock out these two genes, it's lethal in the fetal stage. But what's beauty about zebrafish is they're viable. And there's no problem. You can grow them, you can reproduce them. So we have this ubiquitous GR and MR knockout model in the lab, and we can do all sorts of studies with it. We also have a double knockout, but they lack both GR and MR. So it begs the question, you have these two important receptors being knocked out, and yet the animal can survive. So this comes back to the same sort of idea that Professor Balram talked about, animals find means to survive. So we don't know how they do that, but that's something we are trying to understand in the lab right now. Okay, so now you have this sort of idea that, cortisol is being produced in teleost, which is over here. And most of the teleost underwent another genome duplication event. So they have two copies of every, or multiple copies of every gene. And in zebrafish, that's why it's another good model for knockout studies. They lost their other genes. So they only have one MR and one GR, which is very similar to what we have. So again, from a biomedical stress research standpoint, this becomes a really, really good model. The other thing is MR has a much higher affinity for cortisol than glucocorticoid receptor. So it's 10 times more specific. So when you have low levels of cortisol, all your MR receptors are bound. Whereas when cortisol level increases with stress, that's when your glucocorticoid receptor gets bound. So you have the same hormone controlling two different things with two different receptors, which is really, really interesting. So that's something we were trying to figure out. What exactly is the role of MR under resting conditions? So under resting conditions, your cortisol level is low, but all your MRs are bound, 80%. And then when you stress the animal, cortisol level starts going up, and once it goes above a certain threshold, your GR gets activated, and your MR is already active. So we were trying to figure out what exactly the difference or the distinct response is based on these two receptors. And we know already that whenever GR is active, it plays a very important role in nutrient mobilization, like I mentioned earlier, to provide energy resources, to provide metabolic fuel for your cells to cope with stress. But we have no idea what MR is doing. So that was the focus of Erin's thesis for her PhD work. And so we've tried to figure out exactly what MR is doing and what is its purpose during a stress response and recovery from a stressor. So the third part will be, I'll give you some results from the lab, and we need, it's nice to see the kind of work we're doing, so if students are interested, you can always contact me. So when you do knockout studies, it's good to have a really nice readout of your knockout. So we use behavior as a really neat readout of the responses we see in the knockout model. And with zebrafish, they're really, really tiny. Again, zebrafish is endemic to India, so it's always, when I talk about zebrafish, I always, say this is present in India, mostly in northern India, but it's in every biomedical lab in the world. We have this as a tool now for developmental studies because it's genetically tractable. It's such a great model. So the other nice thing about zebrafish embryo and larvae is you can put them in a 96-well plate. So you can get multiple readings at one shot, and you can put them in behavioral systems and monitor them over 24 hours or 48 hours, and you can do light and dark, you can do circadian studies, all those do that. So that's a system we have. We have a Noldes behavioral system where we can put the zebrafish larvae in all of these wells, and then we found out that there is a really nice behavioral paradigm that we could use. Every time you shine a light on them, they freeze at that particular age. This is 4 days after fertilization. And then you take them in the dark, they become hyperactive. And that's repeatable. So you sort of see like that, you can see them in the light, they freeze. In the dark, they become hyperactive. And you can plot that. I'm sorry, I don't know what happened to the axis and the legend, but it's very consistent in the response. So we use this as a model to develop stress-related behavior or stress-related hyperactivity. And then what we do is, so we validated this as a stress-related behavior. And then next question was, what happens when these guys are stressed? So we treated them with cortisol. We elevated the cortisol level with the idea that glucocorticoid receptors are activated now. And then you can see, what you see is, when you treat them with, sorry about this, I don't know what happened here, but that's cortisol-treated group and that's the control. So every time you treat them with cortisol, they become hyperactive. So this was a really interesting model to see that cortisol is doing something to the animal that makes them hyperactive. But then it begs the question, which receptor is involved in that? Is it GR or MR? We have no idea. So we decided, so that's where the knockout model comes into play. So we did the study with the knockout model. So we had the glucocorticoid receptor knockout and the mineralocorticoid receptor knockout. And this was a real novel finding for us. Both the receptors are required. If you delete one or the other, you don't see this hyperactivity. So that gave us this idea that these receptors don't work in isolation. There is a lot of interaction between these two receptors. And so when you plot it, that's sort of the total distance smoothed. You can see them being hyperactive with cortisol, but in the absence of those receptors, not just GR, but also MR. So the idea is that, and we have proved this. We have done several other studies to confirm that's indeed what's going on. But we don't know what the specific mechanism is. So now what we're looking at is whether is it homodimerization of the receptors independently or just GR and MR form heterodimers, and that's what's driving this sort of behavioral change. So the third part is metabolism and growth. I'll go quickly through that. Again, because we have this model, we have this knockout model, we can manipulate which receptors are being activated by treating them with cortisol, okay? Because always remember, GR gets activated only with cortisol, MR gets activated when the cortisol levels are at the resting levels. So by manipulating these knockout fish with different levels of cortisol, we can selectively activate the different receptors. I won't go into the details, but I think you get the point. And then we have markers to confirm indeed that's the case. Like 11-beta HSD2 is a glucocorticoid responsive gene. So wherever GR is active, you'll see an upregulation of that particular transcript. And similarly, we discovered that leptin is a really good readout for MR activation. So now we are using these two as our readout for GR and MR activation. So then what we found was that one of the key findings was that when fish are treated with cortisol and only in the presence of MR, there is increased lipid accumulation. So we did a RNA-seq transcriptomics. We found a number of pathways all related to lipid metabolism being upregulated in the presence of MR activation. Again, I won't go into the details, but what this is showing you is some of the target genes that are being regulated by MR activation. The red are the genes that are activated by glucocorticoid receptor, and the green are the ones that are activated by mineralocorticoid receptor. So the bottom line is, when MR is activated under resting conditions, there is an increased accumulation of lipid or a decrease in lipid breakdown. It could be either way. Again, I won't go into the details, but if someone is interested, I can talk about the Venn diagram and what the transcriptomics tell you. But what the key finding is, so if you do triglyceride, this is a good marker of lipid accumulation. And when you look at the triglyceride, and that one is showing you lipoprotein lipase transcript. As with MR activation, you see an increase in triglyceride concentration in the larvae. And concurrently, you see a reduction in the breakdown enzyme, the lipoprotein lipase. So this is really cool. So this is sort of the paradigm we use to show that indeed MR is lipogenic in zebrafish. It's a hormone as an adiposity signal which accumulates lipid and reduces lipid breakdown. So MR is anti-lipolytic, and the other interesting finding was this: there is quite a bit of interaction between the two receptors. Between when you have MR-GR, you have a completely different response as opposed to only MR or only GR. So those are the two things we are focusing in the lab right now. How are these interactions occurring, and what is the purpose of these interactions? So I'll just quickly go through this. So then obviously the lipid, there is lipid accumulation, then the natural thing to ask is it affecting growth? So we try to look at growth. I don't know if someone recognizes. So she did her master's here at Kerala University or Central University of Kerala. She finished her master's and now she's doing a PhD in my lab. And she's actually in the third year of her PhD program. So her project is trying to look at the interaction of glucocorticoid receptor and mineralocorticoid receptor and trying to figure out what the phenotypes are associated with those receptor activation. So what we do is we feed the fish 25% body weight to get them to grow faster. So we have a model where we have 5 minutes? Yeah, so then, so what happens is if you have a knockout model, they don't grow at all. So either these are MR knockout. So if you take out MR, they don't grow, they don't accumulate lipid. And we have done several studies, and you can look at the triglycerides. Without MR, you don't have any triglycerides. We don't have any total protein. So the growth is affected in the absence of MR. So what we did was then we thought, let's try to rescue it. So we did micro-injection of the plasmid with MR in it, okay? And we confirmed that indeed, when you add MR plasmid, we could get the readout. We can get an increased MR responsive signal. But what's really neat is when you put it back in, so these are with empty plasmids, and these are plasmids with MR, mineralocorticoid receptor, and you can clearly see the difference. So with MR, you start, it's significantly higher, and that's significantly higher. So what's that telling us is indeed MR is really critical for growth, lipid accumulation, protein synthesis. So the bottom line is, when cortisol goes up, it's energy breakdown mode, quick mobilization of energy resources, but when the animal comes back to the resting level, the recovery phase, MR may become really, really important. So the bottom line is, we did a number of different studies. Again, it's all published. What we found was that under unstressed resting condition, your cortisol, it's at the resting level, MR gets activated, and what MR is doing is increasing uptake of nutrients, it increases storage, and it reduces utilization. On the other hand, when the animal is stressed, MR is sort of silent because GR takes over, and the role of GR is, it reduces the uptake, reduces the storage, increases the breakdown to provide substrates for gluconeogenesis, oxidation, etc. So the working hypothesis right now is that, under resting conditions, MR is active. It has an anabolic role in the animal. And as the cortisol level increases, you have both GR and MR coming together, involved in stress-related behavior and stress-mediated energy repartitioning. And once GR gets active for long periods of time, you have catabolic process occurring. But the key point here is that the recovery requires MR and its anabolic role. So I think that's sort of the take-home message. So I'll quickly go through this. So this is what we're doing right now in the lab. We are trying to figure out what the mechanisms are at the molecular level. We're looking at the interaction of these two receptors. And this is where we are doing a lot of, I have a student right now doing crystallography, trying to figure out exactly what the binding sites are and how these sorts of interactions are taking place. So the physiological role of MR, it has a distinct and complementary role with GR in nutrient homeostasis, growth metabolism, stress-related behavior, stress-mediated energy mobilization. I didn't talk about any of this. It also interacts with insulin. I won't get into that right now. So the bottom line is, as a take-home message, if you don't remember anything, just remember that both MR and GR are essential for stress adaptation, and the tissues, that's our working hypothesis right now. The tissue-specific ratio of GR and MR may determine stress resilience in animals. I have a fairly large lab, and they all do the work. I get to travel and give my talk, so I really need to acknowledge them all. Erin did, she was a PhD student, now she's a faculty in Netherlands, and she did most of the knockout work and also the lipid work. Mira's right now doing the work on interaction of GR and MR. And these are all the funding agencies. They've been very generous in funding my research. Thank you.
Thank you, Professor Matt Vijayan, for this excellent talk, enlightening us on cortisol and MR-GR interactive plays. I can permit just one question for the time constraints.
Yeah, it's wonderful talk, Rajesh here, and definitely fascinating with the cortisol-dependent, stress and then so on. So, and just looking at parallel the mammalian system, when we have the AMPK parallelly acting on this cortisol, do we also find similar kind of paralog or ortholog of a mammalian AMPK with the cortisol-dependent mechanism, or is different?
No, that's different. That's probably a different signaling pathway altogether. Most likely, like insulin activation or something may become important because they do interact. MR actually interacts with insulin. Even GR does, but MR does more than GR does. But that could be a different pathway. We haven't looked at that yet.
Thank you.
You can talk to Professor Matt Vijayan in the lunch due to want of time. Please excuse me. Now I thank you, Professor Matt Vijayan, for your
Thank you. I can talk with you during lunch.
Thank you, sir, for this truly insightful talk and for sharing your expertise with us. Now I request Dr. B. Balasubramaniam to kindly present a memento as a token of our gratitude.
One more thing. I start or they will say something? Yeah. So it is my great pleasure to invite my very good friend, Dr. Shrabani Mukherjee. Dr. Shrabani is an eminent scientist and works on understanding the pathophysiology of PCOS, and she has wonderful awards, publications, and very interestingly, she is going to talk not about her work, but on somebody whom many of the students don't know, Dr. Subhash Mukherjee, and we all reproductive biologists actually feel very this thing that it was India's lost Nobel. So hear about it by Dr. Shrabani.
So today I will be talking about A Life Ahead of Time, Dr. Subhash Mukherjee, the IVF's forgotten visionary. So this is a very close to my heart, and Indrashi specifically requests me to talk about that. And as an Indian, we all should know about his contribution to the world IVF. And I won't be sharing any of my research, but this is more important, and this is aligned with the National Science Day. So can I get a mic? Collar mic? We all know that infertility affects millions of couples. And here, there are a lot of many treatments available for the infertility. And assisted reproductive technique is one of the most important one, which includes IVF, that is in vitro fertilization, that is fertilization in a glass plate, what is called, and IUI, etc. So, even in the mythology also, we have even in mythology also, we know that in Mahabharata, Gandhari, just a minute. So I can move a little bit. Gandhari gave birth to the lump of lifeless bird with sage Vasa. He divided into 101 pots and put with ghee and a nutrient, enriched nutrient, and we give rise to 100 Kauravas after 2 years. So, can I move? Yeah, I can hear. So, 100 Kauravas after 2 years. Slides are not moving. Yeah. So, this can be compared to the modern concepts like IVF, embryo culture, and artificial wombs. But the IVF is very modern technology will develop, which evolved over decades. Mainly from 1950s, the work started where low success animal experiments were carried out till the landmark discovery of the birth of the first IVF baby. So, in 1987, Schenck tried first IVF attempt in the guinea pig and rabbit, but it was all unsuccessful. The first important embryo transfer experiment reported in PNS by Hips, where he transferred the fertilized ova between two breeds of rabbit from Angora rabbit to the Belgium here, which leads to the birth of 6 offspring, which proved that surrogacy is possible. Then Albert Brattet in 1930 successfully did the in vitro culture of pre-implantation rabbit embryos. JBS Haldane, who was a British scientist, he given a lecture in the Cambridge and introduced the ectogenesis, that is development of the whole human being outside the body. It is in the artificial environment. Then comes to the work of one of the pioneers in IVF, Dr. Pincus, who along with Ensman first reported in PNS the in vitro maturation of the ovum, then fertilization and the live birth of the rabbit. He also over a period in 1935 and 39 showed that spontaneous resumption of the oocyte is possible in vitro. But there was a lot of encouragement and lot of controversy, and because of this controversy he has to leave the Harvard and he moved to work on the hormone experiments, and he was the first one to develop the oral contraceptive pill, and he showed that progesterone acts as an inhibitor of the ovulation. Inspired by the work, then John Rock, he is a gynecologist in Boston. He started working development of the human embryo and IVF. He hired Miriam Menken, who used to work with Pincus, and they tried to start human IVF for the first time. So John Rock used to do the surgery, and Miriam used to take the ovary, run to the lab, extract the ova, and used to set for the fertilization experiments. So over 6 years she tried. She 138 fertilization experiment he set up from the 800 embryo. One day he was very tired, he slept off, so he kept the sperms for a longer time, that is 1 hour with the ova. And that day she saw the fertilization for the first time. So, and she could repeat her experiment over few times in the next years. So 1951 is another landmark where both M.C. Chang and Colin Austin independently discovered the process of sperm capacitation. 1961, Palmer and his colleagues Klein was the first to report that laparoscopic retrieval of the oocyte. In 1973, Shetus first tried the IVF pregnancy. He combined the sperm and oocyte of the Doris and John Dell in a test tube. But however, his boss, Dr. Willie, opened the tube after 4 days, contaminated the fertilized material, and that experiment was spoiled. And after that, he has to lift, Shetus has to leave the university, and he moved on. In 1968, David Wattingman first achieved the IVF of the mouse. And first IVF pregnancy in 1973 reported by Carl Wood and John Lytton in the Monash University in Australia. But unfortunately, the miscarriage occurs before 1 week. So Robert Edward were working on this in vitro fertilization since very systematically from 1950s. In 1965, he with his colleagues first attempt to fertilize the human embryo outside the body, and they could achieve 2 cell stage embryos. But by that time, Edward realized that in vitro maturation of egg is not going to give good results, so he thought it is important to get the oocyte just before ovulation, and that will help to fertilize the oocyte. In the meantime, Dr. Patrick Steptoe developed a laparoscopic method to extract the oocyte from the ovary, and he used also the hormonal regimen for the superovulating the women. So both of them, they joined hand, and after that in 1969, they reported the first successful fertilization of human embryo outside the body in the Lancet paper. That gave hope to millions of people. After that, they could create 16-cell embryo, and 1976 they published first pregnancy by IVF, but it was unfortunate the pregnancy was ectopic. So after 13 weeks, they have to terminate the pregnancy, but it gave lot of encouragement to them. And by the meantime, Edward realized that the superovulation, there is a decrease in the luteal phase, the low in the progesterone, which is leading to the miscarriage. So he realized that the superovulation is not going to work. Let us go to the natural cycles, where he started measuring the urinary LH during the normal cycle. In the meantime, Leslie and John Brown was trying to conceive for the last 9 years after the marriage, and they were referred to Dr. Patrick Steptoe, and they explained that they are planning some new experiments. If they agree, they will try for the IVF. So Leslie undergo laparoscopic oocyte retrieval during her natural cycles. Edward fertilized that oocyte and transferred the 8-cell embryo, and on July 25th, 1978, we know the world's first test tube baby, Louis Brown, was born in England. and they published their work in the Lancet paper. But after their landmark discovery, this Edward and Steptoe also faced several skepticism. They were also harassed. They lost all their funding from university, from any funding agency, from hospitals. So they were forced to leave the hospital to establish their first private IVF clinic, Bourne Hall, in 1980, and help many couples, because they realized that this is going to help many couples to get their baby. So there was ethical constraint, so much that Edward has to initiate legal action against British Medical Association and the press to stop them from sabotaging his work and reputation. But still they could publish their work in several journals. Many people supported them, though there was lot of offense, but many people supported them, mainly patients, and from the very rich private organization, they supported their work, so they could continue. Edward got Nobel Prize in 2010 in the physiology of medicine, and that by that time 12 million babies were born to the IVF worldwide. Now coming to the second part, only after 67-day birth of the Louis Brown, India also amazed with the news that a test tube baby born in Calcutta Bellevue Nursing Home, Durga, which was the work of the physician, scientist, clinician Dr. Subhash Mukherjee. He was supported by a team, that is Dr. Sunit Mukherjee, who was a cryobiologist, a professor in food and biochemical engineering in Jadavpur University, and the gynecologist was Saraj Kanti Bhattacharya. So Durga was born out of the cryopreservation, because Dr. Subhash Mukherjee also realized the same thing, that superovulation leads to the reduce the luteal phase. So there is low progesterone, there is a chance of miscarriage. So Sunit Mukherjee and Subhash Mukherjee, they developed the method of cryopreservation of the baby. So I have heard from Sunit Mukherjee that they have done all this in their a small apartment of Subhash Mukherjee in his flat. He made one clinic, and without any sophisticated instrument, just incubator and very small equipment, they did all this, and they studied manually to put the liquid in alcohol, the dry ice, to reduce the temperature by 0.5 degree, 1 degree, how to cryopreserve the baby, because that time like slow cooling method was not available. Whatever they did manually. Then they put, after fertilization, they put the embryo in a glass vial with DMSO, and they freeze that for the 53 days. In the next cycle, when all the hormonal balance were okay, the parents were Prabhat and Bela Devi. When all the hormonal balance were Bela Devi were okay, then he transferred the embryos to the uterus, and he himself has the capacity to measure all the gonadotropins, LH, FSH. He knew how to measure this. He used to do by himself. And actually this this couple, Prabhat and Bela Devi, were referred to Dr. Mukherjee in 1974. He told him that he is going to try some new method, and it is quite possible the child, if it born, they may not be, he or she may not be normal, but they were so desperate, they wanted their own child. So they told, no, they are okay with that. Anyway, they want a child, so please go ahead with that. In the meantime, Dr. Subhash Mukherjee was transferred to the Bakura Sammilani Medical College, that is far away from Calcutta. It used to take 4.5 hours. So what he used to do, every weekend or during holiday, he used to come and do the work. So that's why this work got delayed for a few years. So this is Subhash Mukherjee, this is Saraj Kanti Bhattacharya, and this is Sunit Mukherjee. Here you can see this is the incubator and petri dishes what they use that time. So there was immediately lot of response in the media. DD News air, there's and All India Radio also aired the news of the birth of the first crab preserve baby on 4th October. Ananda Bajar, Jugantar and Patrika, all this news from in the West Bengal. The CBS News sent an envoy to interview Dr. Subhash Mukherjee and his team for a documentary, History of Reproductive Engineering, on 6th December, and they aired this in US. Japan leading newspaper Asahi Shimbun interviewed him and published an article. In 1979, Kyoto University invited the whole team to go there and teach them the IVF, but because of the other reason they could not go. And many couples from India and abroad were highly motivated and they were very happy there is a chance to get their own child. So if we see the background of Dr. Mukherjee, he born in 1931. He obtained BSc degree from Presidency College, Calcutta, then MBBS, National Medical College. He first did PhD degree in the Physiology Department, Presidency College. 60 got married, and from 61 to 63 he went to Edinburgh for his fellowship, Columbo Fellowship, where he did another PhD with John A. Loren, where he worked on the gonadotropins. Then 63 to 67 he was a research associate and visiting lecturer at Edinburgh University Medical School. He also worked in the Royal Infirmary, and again, then he came back to India. He joined as a lecturer, reader and professor of physiology of Nilratan Sarkar Medical College, Calcutta. Then he got transferred to the Bakura Shamilani Medical College, and in that time the IVF baby was born. And in 1980 he transferred to Arjikar College and 81 Regional Institute of Ophthalmology. And in 19th June 1981 he ended his life after all the harassment and insult he got from the fellow clinicians, scientists, the authorities and bureaucrats. So whatever he did, his experiment was much far better than the London counterpart, because he used controlled ovarian hyperstimulation to increase the number of the egg to using the HMG. Because that time HMG was not available in India, he used to ask his relatives who used to go abroad to get the HMG for him. I have seen the handwritten letters of him, and when there is a patient who can afford to get the hormone, he used to ask those patients to get the hormone for the treatment from abroad. He used the transvaginal approach for ovum pickup, because he realized that when you superovulate the ovary, it falls on the back part of the uterus, and from the back posterior part of the uterus you can pick up the ovum, that is colposcopy. Because that time laparoscope was not available in India. So he invented this method to pick up the ovum. Then he cultured the thing in a petri dish without any sophisticated instruments, anything like that. And he developed the method how to freeze and thaw the embryo. Actually, the Edwards, they also tried to do the same thing, but they didn't succeed. They developed the method how to freeze and then thaw. After that, they got the live embryos. And his methods were very cost effective and very innovative. And nowadays, if you see across the world, they follow the same method. It may be have modified, little sophisticated, but most of the time the embryos are thawed and they are transferred in the next cycle. And everybody uses superovulation, we know that. But after doing all this innovative work, he didn't get any encouragement or any praise for that. Initially, he could disseminate his research to some of the scientific meeting for the month of November in 1978, like 5th International Congress, Hormonal Steroid. Then he was interviewed by Dr. B.B. Saxena, Professor of Biochemistry and Endocrinology, Cornell University, then Dr. G.P. Talwar of AIIMS, and then Kenneth Ran of the Professor of Gynecology, Harvard Medical School, and which was live broadcast in Doordarshan on 3rd November. He presented his work Gurgaon University, then hospital, BHU, Benares Hindu University, and different societies. And they could only publish a part of their experiment, how to freeze the embryo in the Indian Journal of Cryogenics. Because after that, there was a lot of problem, because there was hue and cry regarding the truthfulness of his experiments. People didn't believe that if it is possible to do such a great experiment in a low budget setup without any sophisticated instrument. Nobody believed, and there was a lot of skepticism, then a lot of clinician also after him. Then it evoked the government. So government set up an inquiry committee, and if you see the constitution of the inquiry committee, composed of the radiophysicist who was a chairperson, then gynecologist, physiologist, nuclear physics, and none of them has really experimented to understand what work he has done. The committee ultimately said that the work is absolutely bogus, the work is fraudulent, and it was also announced by the health minister Nani Bhattacharya in 1978, and they were not allowed to write any scientific paper, communicate with any journal, go to any conference to present their work. So his hand was really tied up. He couldn't share his work. It's such a groundbreaking work with anybody in scientific community. Between all this, the Agarwal family was a very orthodox Marwari family, because they got scared that if this news come out, they will be harassed by their own family. So in between, they also sent legal notice to Dr. Subhash Mukherjee that you can't reveal anything about the IVF. So Dr. Mukherjee's hands was tied from both the sides. So this is the inquiry committee. You can see who are the members of the committee. So what happened after all this humiliation, insult, ostracization? He was very upset. In 1980, he had a myocardial infarction. But still he has to travel to Bankura, it's very far away. So he requested the health minister to transfer him to a medical college in Calcutta. He was transferred to RG Kaur Medical College in Calcutta, but it was in the first floor, so again he has to climb. His health was not good. So then he asked for again transfer to some medical college where he can have his office in the ground floor. So, but then he was finally transferred to Regional Institute of Ophthalmology as a professor of electrophysiology. This killed all his hope to do any IVF research, because the head of the department also told, I can support you if you are willing to do any other research, but not IVF, because the authorities have told not to work on IVF. So this killed all his hope, and after that he committed suicide, and he wrote in the note, I can't wait for a day by a heart attack every day. So we lost a great scientist. He was very curious, innovative, and he used to think out of the box. He has his own, in NRS, he used to stay in the first floor, and the ground floor there was an empty flat, so he transformed that in an animal house. So he has rat, rabbit, guinea pig, monkey, everything. And I have heard that he used to spend most of his salary spending money on this animal house. Even in his department of physiology in the medical college, he created the research environment, he started research activity, and he has many MD and PhD students who worked with him, and he was very courageous. He used to support students like anything. So what he pioneered in which aspect, later on as he could not share with the world his work, so later on many other people, scientists, they claim as the first one. Like first IVF baby following oocyte retrieval from stimulated ovary, Jones in 1981 published. Delayed oocyte insemination, Tronson in 1982. Then first frozen thawed embryo transferred, again Tronson in more 1983. First vaginal approach to retrieve embryo, Torod in 1986. So his name is nowhere actually, if you see. But all this method, he is the pioneer. He developed this method. So after his death, Sunit Mukherjee was a very good friend, means awesome friend of Dr. Subhash Mukherjee. After his death, Dr. Sunit Mukherjee, along with Dr. Subhash Mukherjee's wife, Namita Mukherjee, they tried with different authorities, different people to give the recognition to Subhash Mukherjee, was they are unable to do that. By the meantime, they established a Subhash Mukherjee Memorial Reproductive Biology Research Center in Behala in Calcutta with the help of the Indian Cryogenic Council located in the Jadavpur University, because Sunit Mukherjee was a member of, one of the pioneer member of Indian Cryogenic Council. They used to organize memorial lecture on the birthday of Subhash Mukherjee every year, and they used to call eminent scientists across India to give a talk. So here you can see this one is the establishment of the Subhash Mukherjee Memorial Center. Then these are the different talk. Here you can see this is Dr. Mahale, our former director in IRCH. He also gave the oration. This is the few of the other Subhash Mukherjee's photographs, wife with Namita Mukherjee and with Sunit Mukherjee. So after 8 years, in 1986, at our institute, I belongs to NIRCH, it was earlier name was IRR. So at our institute, under the work of Dr. T.C. Anand Kumar, who was that time the director, and Dr. Indira Hinduja, who was a gynecologist at the KM hospital, the first scientifically documented IVF baby birth occurs at our institute. That is the baby Harsha. Here you can see the baby Harsha with Dr. Hinduja holding her, a baby Harsha and Dr. Hinduja at the present time. So when the news broke up, it was very appreciated, everybody liked, and they could publish their paper in the JAG. Everything was very well accepted. Can I get some water? Any accepted? What happened in 1996, T.C. Anand Kumar were invited to deliver a talk in the Subhash Mukherjee oration lecture. Fine, I will cover. Thank you. That time, Dr. Sunit Mukherjee requested to go through all the notes, handwritten notes of Dr. Subhash Mukherjee, and after going through all these notes, Dr. T.C. Anand Kumar realized that yes, Subhash Mukherjee really indeed did the first IVF baby of India, and Dr. T.C. Anand Kumar was such a generous person. He gave his crown to Subhash Mukherjee. He first published an article in the Current Science where he published the work of the Subhash Mukherjee. And he was after ICMR to recognize Dr. Subhash Mukherjee's work. So in 2002, ICMR also recognized the work of Subhash Mukherjee as he was the first creator of the first IVF baby, and the creator of India, and creator of the first frozen IVF baby of the world, and second IVF baby of the world. So in 2003, Durga, that is Kanupriya, her name is Kanupriya, was first introduced to the world during the Silver Jubilee of the IVF at a conference in Bangalore, which was organized mainly by Dr. T.C. Anand Kumar. Here you can see T.C. Anand Kumar with Dr. T.C. Anand Kumar with Kanupriya. So after that, there was a lot of media response that he should get Bharat Ratna, his work should be recognized, and lot of things came. So even many of you have seen this movie, like Dr. Ki Maut by Tapan Sinha, this is based on the life of Dr. Subhash Mukherjee, but he just changed it to the development of a vaccine rather than IVF. And after this, Dr. Subhash Mukherjee got recognition from many of the he got honorary DSc degree from Indian Statistical Institute. ICMS introduced Dr. Subhash Mukherjee Award. 2 chairs named after Dr. Mukherjee by West Bengal government. One is the Physiology Department of NRS College and another School of Tropical Medicine. And post of the professor in the physiology named Subhash Mukherjee, Professor of Physiology in NRS Medical College. So this is the Nobel Prize for the IVF baby, first IVF baby conferred to Dr. Edward. If Subhash Mukherjee was there, he could have shared the prize and India would have got the first Nobel Prize in the medicine and physiology. So because of our own people, we lost this prize and we lost this great scientist. So I will just move fast. Apart from the IVF, he was a very renowned endocrinologist, physiologist, and he was very skilled surgeon. So he has worked on biochemical changes and hormonal pregnancy in the abnormal pregnancy in ectopic toxemia. He developed a very sensitive LH method of ovarian cholesterol depletion, which was 5 million times sensitive than the earlier LH assay, which used to ovarian ascorbic acid depletion method. And he helped many other people in many other aspects. So other than this is one of the important thing, he is the one who started working using androgen to treat the female reproductive problems. So that time he studied that effect of testosterone and DHEA on the rat model and observed that luteotropic and uterotropic effect, which increases the weight of the uterus and the ovary. And DHEA treatment increased the number of the follicles in the PMS treated, and nowadays in the IVF, poor ovarian responder, they give DHEA treatment for that. And he also treated the patient with luteal phase insufficiency with the testosterone in that time. Then I work on PCOS mainly, so that time he developed the PCOS model and he did unilateral oophorectomy, restored fertility in them. Then he carried out bilateral wage resection in the ovary of the woman and he observed that there is a restoration of the gonadotropin and steroid. He hypothesized that in that era that stress is one of the reasons for the PCOS. Because he had several patients with PCOS and he used to take a lot of detailed history and he realized all this patient has very chronic stress. So he noted this. He published this also that PCOS, that stress through adrenocorticoid, ACTH and all this ACTH and androgen induce the PCOS. And I will just feel he has a contribution in the field of nutrition and some other thing. I will just take 1 minute to talk about Dr. Sunit Mukherjee. He was a very good friend of Dr. Subhash Mukherjee. So after death of the Subhash Mukherjee, he tried with different people, different organization authorities to restore his fame. And he kept all his documents with him in his that Subhash Mukherjee Memorial Center. So what happened in the figure, you can see in this picture, Sunit Mukherjee with Rajvi and myself. So one of the meeting, he met Rajvi. Rajvi is one of the first student of the Dr. T.C. Anand Kumar. He told that, see, I have so many things of Subhash Mukherjee, but I don't know after that what will happen if ICMR can take care of that. In that way, we were introduced to Sunit Mukherjee and I have a project of INSA to organize all these things. So this was this is the place where all his documents were there. So I have visited several times and took team and got record of all the documents, whatever was possible, but they are all dilapidated in a very good back condition. These are the books. Here you can see a paper by Indrashish also on Subhash Mukherjee. And we released a booklet on Subhash Mukherjee, and it was released by our Union Health Minister, Dr. Harshvardhan, and ICMR DG, Dr. Balaram Bhargav that time. It is in our website. I urge all of you, please read that book, because we should know about Subhash Mukherjee. And this is my last slide. Thanks to all of you. And this is the drawn by Dr. Subhash Mukherjee. This is a Madonna. I draw picture of live Madonnas with hormones. This is the mother with child with hypothalamus, pituitary, ovarian axis. He was not only scientist, he was admirer of art, music, and literature also. He used to able to speak 4 languages. He used to recite Gita like anything. He was a very classical singer also. So thanks.
Talk. There will not be any questions because it's a tribute. It will be nice if everybody stands up for a minute to give homage to Professor Subhash Mukherjee, and I think that will be a fitting. Thank you so much.
So I'm very happy that aligned with the National Science Day.
Thank you, ma'am, for the truly insightful talk about Dr. Subhash Mukherjee and IVF's forgotten visionary. I now request Dr. Malini Laloria to kindly present the memento as a token of our appreciation. And I extend my sincere gratitude to Chairpersons Dr. B. Subramanian and Dr. B. Malini Laloria for your time and coordination. Thank you, sir. Thank you, ma'am. Thank you. We will take a lunch break. Lunch will be served outside the hall. All are kindly requested to reassemble.